Hey, so now that we know all about mitosis, let's turn our attention to the next set of details is meiosis, or sexual reproduction, sexual cells. And so it doesn't matter if you have the student notes or if you have the PowerPoint pulled out. They're the same thing. So go ahead and pull that out. For the video, I'm gonna use the PowerPoint images, but think about meiosis. So mitosis, remember, was diploid cells that reproduce and make identical diploid cells, right? So mitosis had prophase, metaphase, anaphase, telophase. It made two. That 2N means diploid cells, two identical diploid cells. And this was like for skin cells, bone cells, growth, repair, and even developing the baby from the zygote. Meiosis is different. Meiosis is where there's actually two divisions. You can see here that's one cell divided into two, and that's two cells divided into four. It makes four haploid cells. Haploid are cells that only have 23 chromosomes. They don't have pairs of chromosomes. So, and there's two divisions here. So let's jump in. Right off the bat, diploid refers to a cell with two of each chromosome, where haploid is half of each chromosome. Another word is somatic or body cells, gametes or sex cells. So make sure you know the difference. A diploid cell is a somatic cell, and a haploid cell is a gametic cell or a gamete. The only haploid cells in humans are sperm cells or egg cells. Of course, you know that females have eggs and males have sperm. So remember, diploid has pairs of chromosomes, 23 pairs. Haploid doesn't have pairs. It only has 23 chromosomes. Somatic cells are diploid. Gametic cells are haploid. Okay, you're ready. You got it. Make sure you know that whole slide. Okay, meiosis is... this allows zygotes to have the same... So when the sperm and the egg go together... Yeah, we should know that for sure. When the sperm and the egg go together, it makes one diploid cell. So the sperm is haploid, the egg is haploid. When it goes together, it makes one diploid cell. Okay, so meiosis it says right here, has two divisions. It's where a diploid cell reduces down to a haploid cell, right? So a type of nuclear division that reduces the chromosome number from diploid to haploid. Okay, highlight, underline, reduces, here we go, reduces the chromosome. Okay, homologous chromosomes that's the pairs. HOMO means same. And so homologous chromosomes are chromosomes that contain genes for the same trait. This does not mean that the gene codes for the same version of that trait. So you have like one for mom and one for dad. And chromosome number 1 has the same, like, you know, just for example, like if your earlobe, what your earlobe looks like on chromosome number 1, one from mom and one from dad. That's homologous chromosomes. Okay, an allele is an alternate form of a gene. So you can see there in the picture, the allele occurs on the same location, or homologous chromosome. So the allele is that this little tiny segment right here, this section, this band. That's the allele for purple flower, and this is the allele for white flower. Okay, in meiosis 1, a diploid cell divides, and then in meiosis 2, it divides again. So there are two divisions, is what that's saying. Okay, let's look at this, and you want to highlight, it's already bold right here, but highlight homologous, in meiosis 1, the homologous chromosomes line up, like in metaphase. Okay, so meiosis 1 has prophase, metaphase, anaphase, telophase. Meiosis 2 has prophase, metaphase, anaphase, telophase. Okay, so in meiosis 1, the homologous chromosomes line up. In meiosis 2, halves line up. The sister chromatids line up. So meiosis 2 is more like mitosis in a way, but don't think about that. but in a way, yes. Okay, meiosis requires 2 divisions and makes haploid daughter cells. 4 sperm or egg cells. 4 different cells, sperm or egg. Okay, just some vocabulary. The synapsis occurs in meiosis 1 when the homologous chromosomes line up in pairs in the center of the nucleus. That's called synapsis. And you can see it right here. They line up in pairs. Okay, bivalent or 2 homologous chromosome pairs that stay in close association. So chromosomes that are generally close together and inherited together. Okay, so let's look at this right here. Like me and my sister have the same mom and dad, and we look totally different. Why? We have the same mom and the same dad, so why do we look so different? Meiosis brings about genetic variation of a species in 2 key ways. The first one is the crossing over. The chromosomes actually cross over and exchange. They switch genetic material between non-sister chromatids of those bivalents during meiosis 1. During crossing over happens during meiosis 1. Specifically, it happens during prophase 1 of meiosis 1. This allows the offspring to have different set of genes. Okay, and then those chromosomes sort independently. Okay, so just because you got your mom's eye color doesn't mean you're going to get your mom's nose shape, or just because you got you know, the one trait from your mom doesn't mean you're going to get another trait from your mom. Now, some are usually together, like freckles and red hair may be usually together, but not always. Some people have red hair and no freckles, or freckles and no red hair. When bivalents line up at the metaphase plate during metaphase 1, their alignment is completely random. This allows many genetic combinations of haploid gametes to be produced. And so my sister and I have the same mom and dad, but we look very different. She has darker hair than I do. She has darker eyes than I do. I'm much taller than her. And that's 2 reasons for meiosis causes genetic variation. The first one is the crossing over. Those homologous chromosomes cross over. And then the second one is the chromosomes sort independently of one another. So this is, highlight all of this, crossing over and independent assortment. Maybe I can show you here. Okay, so in prophase 1 of meiosis 1, the chromosomes will actually cross over. You can see the orange and the red cross over, and now they've switched, like they've switched some of the alleles. And so just because you got the orange, let's say the orange is from the dad, just because you got the orange chromosome from your dad, you got a little bit of your mom's red chromosome. And in different siblings, in different eggs, it may cross over in different places. So for example, you know, right here they crossed over what's looking like the lower part of the chromosome, like, you know, like the legs. They could have crossed over the hands. They could cross over closer to the centromere or further away. Okay, and then look right here, like this is independent assortment. Both of these things create genetic variation. If you're looking at this right here, it's like the red and the blue are lined up. It doesn't really do that. You know, chromosome 1 pairs with chromosome 1, but it's not like all from your mom get on one side and all from your dad get on the other side. So you can see here in this image, some of them are switched. They sort independently. These are different options of how they could have lined up. So when you make, if it makes 4 sperm cells, it depends on, you may look different depending on which sperm cell fertilized the egg that created you, because 2 reasons, crossing over and independent assortment. Okay, in interphase 1, that's the in between. Grow, replicate DNA, grow. DNA replication occurs in that S phase. Okay, so interphase is the same. Interphase is in between, right? That's in between divisions. And let me go back right here and let me show you this right here. If you look right here, there's, you know, prophase, metaphase, anaphase, and telophase in both meiosis 1 and meiosis 2. Okay, so before they divide, it's called interphase, and you can see that here. Here's a picture. This is a plant cell. This is an animal cell. Prophase, metaphase, anaphase, telophase. It starts to divide. There's interkinesis, and then here it is again. Prophase, metaphase, anaphase. This is the cell, and then telophase again. So you can see how the cell has two divisions. Prophase, this is similar to prophase in mitosis. Spindle fibers form. Nuclear envelope begins to fall apart. Homologous chromosomes undergo that synapsis where they attach to spindle fibers. Crossing over occurs here. Highlight, underline, circle. That's going to be a test question. Where does crossing over occur? Prophase 1. You see them crossing over. See how they've crossed over their DNA? They're crossing over DNA segments. Exchange. Let's see. So highlight all of this. Uh-oh. There we go. DNA segments exchange between homologous chromosomes. Metaphase, same thing really. They line up in the middle. The bivalents are held together by chiasmata at the metaphase plate. Okay. Anaphase, this is the difference right here. Look right here. This is the difference between metaphase and mitosis. This one's mitosis. The homologous chromosomes line up in, the chromosomes line up in mitosis. In meiosis, the homologous chromosomes line up. Okay, so these are, in mitosis, the chromosomes line up in the middle. In meiosis, the homologous pairs, the pairs of chromosomes line up in the middle, and then you can see they would start to separate. In mitosis, just the sister chromatid separates. In meiosis, the whole chromosome separates. There's anaphase. You can see the image. Homologous chromosomes, highlight, underline, homologous chromosome pairs migrate to opposite ends. So in mitosis, you wanted to know that the sister chromatids separate. In anaphase of meiosis, homologous chromosomes separate. And there's telophase, you can see there. In meiosis 2, after a brief interkinesis without DNA replication. So we don't need to make any more DNA because we're going from diploid to haploid. After this, it's normal meiotic division. Because there's no DNA replication, we're reducing. Meiosis is a reduction of chromosomes, reduction of DNA. So same thing. The cell membrane breaks down, the nucleolus goes away, spindle fibers form. Each cell has one chromosome from the homologous pair. In metaphase, they line up at the metaphase plate. In anaphase, the sister chromatids start to pull apart. That's what you can see in that picture. Telophase, the cell membrane starts to break apart. You've got that cleavage furrowing again. Okay, so go back. I was looking at my notes. I want to go back and make sure I tell you right here. There is no interphase. There's no, there's no DNA replication here. On metaphase slide, highlight sister chromatids at the centromere. Sister chromatids. Remember, in metaphase 1, it was homologous chromosomes. Okay, and then in telophase, you've got 4 daughter cells that are created. No 2 chromosomes are alike because of the crossing over. So you can see here, like this one's like a big red and a little blue, and a big blue and a little red. You see what I'm saying? No 2 are the same. So you've made 4 haploid different cells. 4 haploid different cells. Now, when the sperm and the egg go together, then the diploid is sometimes, like, books will use DN to represent diploid. The diploid is restored when sperm and egg go together, but this has made 4. That one N means haploid. There we go. 4 haploid cells that are all different. Okay, so here's a picture of meiosis. You can see the chromosomes double right here. You can see these are meiosis 1. The chromosome, this only got 1 chromosome in it, showing you for the image. And then the sister chromatids separate on the second division, and you make 4, 1, 2, 3, 4 different cells. Okay, so here's another image comparing. We just talked about, we'll do mitosis first. Mitosis is 1 division. It produces 2 identical diploid cells, and they're all identical. This is for skin repair, bone repair. Meiosis has 2 division. It produces 4 haploid cells. Haploid cells. They are all different. Okay, so 2 terms I want you to know. The production of sperm is called spermatogenesis. Spermatogenesis. It's really interesting. Sperm cells are produced when a male starts to go through puberty, and they're produced for the rest of his life. New sperm cells, all 4 of them are viable. Stem cells in the testes. They keep testes supplied with the primary spermatocytes. And so it's interesting the difference in how sperm are produced. All throughout a male's life, they make more and more and more and more. And of course, the hormones do impact the amount they make, but they do make sperm all throughout, and all 4 are good. Oogenesis is the production of egg cells. Ovaries contain stem cells. It's interesting. They're produced during fetal development. So when a little girl is born, she has all the egg cells. They're premature, but that's all that she's going to make. So once they're produced during fetal development, so when the female is in the womb, that's when she produces them. So they produce the primary oocytes during fetal development, undergo oogenesis once the female is sexually mature. So they mature once the little girl matures, but she has the starter cells the whole time she's born with them. So primary oocytes divide through meiosis 1 into 2 cells called secondary oocytes and polar bodies. And then you can see right here in the image, no you can't. Yeah. So this right here is showing you meiosis 1, and then once it's fertilized, then you see meiosis 2 is completed after the entry of the sperm. And so it doesn't actually complete the meiosis until after the sperm has entered. Secondary oocytes begin meiosis 2, but stop at metaphase. It leaves the ovary and enters down into the oviduct. No sperm. Then if there's no sperm, then the egg would disintegrate. If there is a sperm, then it continues through meiosis 2, and it forms a second polar body. Mature eggs have 23 chromosomes. A zygote has 23 pairs, 23 from the sperm and 23 from the egg. So it's really interesting that females are actually born with all the oocytes that they're going to make. And that's why the age of the female is significant, because the age of the eggs are also that old. So euploidy is having the correct number of chromosomes, 23 pairs. Aneuploidy is a change in the chromosome number, usually from when those chromosomes sort independently, they didn't sort correctly. Non-disjunction is where the chromosomes didn't sort correctly. One goes to one egg and one goes to the other. If they kind of stuck together, then you would see, like if you look at this image right here, it's supposed to have 2, but the eggs didn't, the chromosome didn't didn't separate correctly. Monosomy is when it only has one. Instead of having a pair, the word monosomy is where the cell only has one of those chromosomes. You're supposed to have two of every chromosome. Trisomy is where it has three. And so we know about, you may have heard about Down syndrome, is where the 21st chromosome, there's three of them. There's three of the 21st chromosome. So Down syndrome is something, and I always talk about this in class. Down syndrome is nothing that is inherited from the mom or the dad. You don't carry that gene. Down syndrome was when the sperm and egg combined, one of them had an extra chromosome. Instead of it being 23 in the egg and 23 in the sperm accidentally had 24, had an extra chromosome, or the egg, either one. And so Down syndrome it's like, I heard a girl one time say that, it's because she didn't take her vitamins, or because, no, it's not. Down syndrome in a child is because the chromosomes fail to separate correctly, and then the zygote had an extra chromosome in every single cell. And so every single cell in your body has an extra chromosome when we talk about what is Down syndrome. The symptoms of Down syndrome, or it's also called trisomy 21. There's three chromosomes on that 21st pair. They're short in stature. Their eyelids may appear differently. They a lot of times are born with heart problems and have to have different procedures for their heart. They can increase of having, if they have Down syndrome, they have an increased chance of having a child with Down syndrome. But what this is saying is, what this is talking about is the age of the woman. And so if a woman, I'd say 35, but your book says 30. If the woman is over age 30, she has the chances of having a child with Down syndrome are greater. And so that's because those chromosomes are more likely to separate incorrectly. If a woman is over 30 or 35, you're more likely to have a child with Down syndrome. And a lot of it depends on the woman's age, because we were talking about males make sperm every day after puberty, where the woman, their oocytes are made during fetal development. And so when we talk about different genetic disorders, it's usually, if they pertain to age, we always are considering the woman's age. And so these are the symptoms of Down syndrome that you may see. The longer the oocytes are stored, like in the woman's ovaries, the greater the chance of non-disjunction. You want to know that non-disjunction is where the chromosomes fail to separate correctly, and it can cause different types. It's not just Down syndrome. Monosomy is where there's one chromosome. Trisomy is where there's three, and some are more serious than others. Down syndrome children seem to be the happiest people, and people with Down syndrome can reproduce, but it is rare. Okay, abnormal number of sex chromosomes. So if you that X and Y is a male, XX is a female. If you didn't already know that, write it down. XY is a male. The Y is real short, and XX is a female. Let me go back to this karyotype. I'll show you. Well, there it is. Okay, if you look right here at this picture, so there's 22 pairs of chromosomes, and then this one is the sex chromosome. This is a male. It's XY. Females have XX. Okay, so what if you get an extra of the sex chromosomes, inheriting too many or too few, also caused by non-disjunction? Okay, one is called Turner syndrome. That's where you have X and then nothing. You only have an X. So it's a monosomy. This was showing you that you have to have an X. If it's just a Y, the baby won't live. Females with single X chromosomes, Turner syndrome, people with Turner syndrome are shorter. They have a broad chest. I actually know someone with Turner syndrome, and she looks very, very typical. She doesn't look atypical at all. Widely spaced nipples. They usually can't have children, can be of normal intelligence, and function with hormone therapy. So with hormone therapy, they can function. Klinefelter syndrome is where you have XX and a Y. So you have 3. This is like trisomy. Male with underdeveloped testes and prostate. So these may have female and male sex organs, longer arms, longer legs, near normal intelligence. No matter how many X chromosomes present, if you have a Y, we consider it a male. Okay, and so here's images of, are these not the cutest children you've ever seen? Turner syndrome with, you can see here, X, oh no, I messed up. Okay, so Turner syndrome right here, they just have a Y. Let's see, right here, I'm sorry, just have an X. Phew. Turner syndrome just has an X. Klinefelter syndrome has XXY. So they have 3. So this is trisomy, where Turner syndrome is monosomy. And then there's a couple of other non-disjunction disorders that people can have. You can see poly X, where a woman has 3 X's. Jacob syndrome is XYY, so 2 of the Y. Remember, if you have a Y, we consider the person a male. Sometimes the genetic disorder is not a whole chromosome. It's just a part of a chromosome. Part of the chromosome could be deleted, duplicated, or moved, translocated. So that's really interesting. This may be just a segment of a chromosome. I hope this was helpful.